This talk is going to be perhaps a little bit different, and I'm going to ask for forgiveness early. My background is not as a toxicologist. I'm actually a small molecule organic chemist. I also have a JD, and I currently work at US Pharmacopoeia. Next slide, please. So, US Pharmacopoeia, what is a pharmacopoeia? We are a nonprofit organization. Our mission is on the screen. That's to improve global health through public standards. And what this essentially means is that we work with innovative pharmaceutical manufacturers to create and enable generic drugs. So if you are a generic drug manufacturer, there is a USP standard that you have to comply with as far as quality, packaging, labeling, et cetera, to be able to market your product in the United States. As a consequence of being the US Pharmacopoeia, our standards are often the de facto regulation in countries that do not have strong regulatory health programs. So we have a worldwide impact, and we use that impact to make quality drugs available and enable manufacturing of those. I did mention we're a nonprofit organization. That's going to be important later. Next slide. What do our standards look like? Well, in 1820, we had a single book. That book has been renamed into the USPNF. That's the National Formulary, and it contains all kinds of fun information related to drug quality. You can see two on the screen here: heparin sodium, which is a large molecule. It needs to have the proper proton spectrum, chromatographic identity, and then you can see filgrastim, which actually is also a large molecule. But most of our national formulary standards pertain to small molecules. Next slide. So you may be wondering why I'm speaking to you today. That is an excellent question. Right now, USP, in support of our small molecule quality program, not only do we create standards for generic manufacturers to comply with, but we also create reference materials that folks can run through their analytical systems to ensure that they are running properly, essentially a validation, if you will. So we have an extensive catalog of small molecules for that purpose. So that's some background. The problem that we're looking at, and I believe this is not new information for you all, but there are challenges understanding mechanisms for small molecule toxicity and consequently the risk that these small molecules may present. Not on this slide includes the new methods for determining toxicity, new methods, new assays, new modes for understanding how these toxicity pathways work. And that's where USP comes in. Next slide, please. So what we envision is repurposing USP, a small portion of USP's existing small molecule catalog, into libraries dedicated to be positive and negative controls for adverse outcome pathways, and specifically the OECD recognized AOPs. So what researchers will be able to do with these small libraries are investigate activity, validate and refine models. They can use these molecules to design and synthesize safer pharmaceutical compounds. And of course, this will increase the understanding of toxicity and increase the amount of toxicity data that's available. So that's the dream. Next slide. Just very briefly, benefits will improve the regulatory uptake of new approach methodologies. With the positive and negative controls, we can demonstrate that these assays and methods are working appropriately. We can enhance predictive toxicology, reduce animal testing. That is something that is very important for USP, and it will be a focus for us in the very near future. Happy to have further conversations about that should folks be interested in that. And as mentioned on the previous slide as well, increase the general availability of tox data. Next slide, please. USP, what do we bring to the table other than a small molecule portfolio, which I'm sure you all are aware, there are many small molecule suppliers out there. What is different about USP? First, we work with a lot of experts in the community to ensure that we have the right samples and materials available for use. We are a company that's 1,200 people large, and I would say approximately 500 of them are analytical chemists. So we have many quality control experts ensuring the small molecules and large molecules that we provide to the community are indeed high quality and capable of performing in the various methods as advertised. The other benefit to the community is that rather than calling up a bunch of material suppliers and hoping they have the materials that you need and the quantity that you need, it's a one-stop shop. So you can just identify an AOP that we have a small library for, and you'll have all of your positive and negative controls available in one spot. Next slide, please. So, and Connie, there's a few animations, so you can go ahead and click through twice, I believe. So, thinking about next steps here, we've got an example library for the TGX DDI biomarker, and the highlighted, one more time, Connie, if you could click. The highlighted compounds, so on the left-hand side, obviously, you can see the positive controls, and on the right-hand side, you can see the negative controls. The compounds that we've highlighted in orange are ones that we already have in the USP's catalog. So what we could do is essentially, as a pilot project, is assemble these materials in a small kit and then make that available for use to explore this particular biomarker. So that's the hope. And just as a side note here, we are working on sourcing benzoapyrene. So we hope to have that as a positive control in this list in the not too distant future. So I do have a couple of questions on the next slide. This proposal really came through a conversation I had with Carol Yawk, and I'm happy to have her chime in with any details that I had missed before we get into the questions for the group. Carol?
Thanks, Brett. I hope everybody can see why I was really interested in Brett bringing this proposal forward. So when we're developing AOPs, we are required to identify the stressors that activate the molecular initiating event. What we're not required to do is to identify the compounds that we should use as negatives. And I would say in MIE development that this aspect of it is not well done. But if we want assays for these molecular initiating events or key events, if we want to develop those methods, we need to have, what are the chemicals that we can use to validate the test method, but also we need this for proficiency testing. So when a new lab is going to adopt a methodology, they typically need to do a proficiency test, demonstrate that they can get the right positives and the right negatives when they need to. And so I was obviously, really excited about the potential of including this, for example, in the TGX DDI FDA biomarker qualification, that not only would we have, a protocol for how the proficiency testing could be done, but we could also have a link to USP where people could, as Brett said, like a one-stop shop, purchase all the QA/QC'd reference chemicals needed to establish that method in your lab. And I could see this applying to a lot of the different methods that eSTAR and other groups, other committees at HESI are undertaking right now. And so that's the driver for me, and appreciate your presentation, Brett. Curious to hear what people think.
Yeah, thanks, Carol. And just maybe to address the elephant in the room here, USP is a nonprofit organization. I think the business development people don't pay any attention to me. This is sort of the science and the small molecule inventory managers kind of being nice to me about this proposal. So, if it receives a warm welcome in the Tox community, I think that would be great, and that would spur kind of more conversations. But that's a long way of saying this is not a big profit driver. I know that's a concern of some folks. I just wanted to mention briefly that this is not really a profit driver for USP, but really is a service to the community and a continuation of our mission. So with that said, just a couple of questions on the screen for folks to think about. Number one, is this interesting to you? Are there other MIEs we should consider? And would you be interested in pressure testing some of these small molecules with us? Happy to have a further conversation about what that might look like. So with that in mind, would love to hear some comments from the audience.
Thanks, Brett. And I think when we chatted, me, you, and Carol, ideas for MIEs might be a good place to start, especially if there's particular ones, because that could help us, find what compounds and what labs and what to do. But definitely open to if folks want to answer any of these.
I see I got the pre-lunch slot, so everyone's blood sugar is extremely low. I'm available via email if folks should want to reach out in a less vocal fashion. My email is not on the screen, but I can give it to everyone, and I'll put it in the chat. It's brett.howard@usp.org.
I think we're starting to get some. So I see Deidre raised her hand.
Yes. Hi, Brett. Thank you so much for explaining all of that and what you're trying to do. I mean, honestly, from my personal opinion, I think that it would be great to be able to have such lists available and make things so much easier. Just having been, that person in the lab trying to go through all the databases and all the suppliers and really trying to figure out, are, they have the right quality? Are they of the right, concentration? Just being able to have that, like as Carol, mentioned, just readily available would save so much time, I think, for across, so many different companies and different sectors that may want to have those. I think that, depending what we're doing, I know they're available. I think that if as time moves on, like you said, if there's different, MIEs, if there's different AOPs that are getting generated, to have, say, smaller lists too, because I know it's not a profit driver, but, depending on the cost, just for example, if I wanted, 20 of the 45 that may be in a panel, I don't need to use quite all of them. So just having a way to be able to potentially have that panel, and I know you said it's available, but be able to select maybe a smaller one. But, for those bigger efforts, I think of, validating different labs and having new folks, I think it's a fantastic idea personally.
Thank you very
Thanks.
much.
Thanks, Deidre. Yeah, one thought I had, I mean, there's the gene tox and we have TGX DDI. Are there other like quote unquote easy ones? So, I mean, we've done a lot of work in carcinogenomics to try and annotate compounds for certain pathways. So maybe that's something we could look into. Another one that comes to mind is all the endocrine work. There's many in vitro assays. Some are on the maybe eco side, but some are for human as well. So that might be another nice source. But I think from this team, what would be of interest? What would be useful for you all, either in your own research or setting up a new assay or, yeah. Yeah, Adam, thyroid is one. I know there's quite a few vitro assays in development and there's some in vivo as well. And sometimes what's nice about those is there are defined lists already. I'm a big fan of not reinventing the wheel. So we could use that as a starting place in some cases, but really want to hear from you all, like, what would be the biggest ones? Yeah, agreed, Carol. So Brett, what I think we can do is, we put your email in there. I think this would be a nice follow-up call to have maybe, a meeting with those that were interested, and maybe we come up with a few ideas ahead of time and bring to the team. So I really asked folks to continue to write either MIEs or pathways that would be helpful to them. And then we can continue to explore, because I think as Deidre articulated, there's a lot of utility for this. Does
I
that
think that
sound
sounds
reasonable?
fantastic. And next time we chat, I won't have this mustache.
That's
So
a nice cliffhanger.
this is going away. Oh, good grief. Anyway, it was good to see you all. Thanks again, Connie, so much for the invitation to chat. Carol, look forward to chatting with you more. And yeah, take care all. Enjoy the rest of your workshop. Thanks.
Thanks, Brett. Thanks, Carol.